Hi, Julie. I'll just start by introducing myself. My name is Dalton Towers. I'm a third-year PhD student here at UT Austin. I'm co-advised between Dr. George Giorgio from the Chemical Engineering Department, as well as Gregory Polito and Andrew Ellington from the Molecular Biosciences Department. And my research focuses primarily on looking at antibody immune repertoires, looking at what type of antibodies we produce when we've been infected or have been vaccinated, and seeing what behaviors they have and how prevalent they are in the serum. And so my research focuses primarily on looking at unusual antibody behaviors. So I look at this feature called polyreactivity in response to influenza vaccinations. And that really is really important story when we talk about universal vaccines, as we try to target many these conserved epitopes for trying to provide vaccines that provide better coverage for new variants. We want to try to understand what type of antibodies target these heavily conserved regions, and polyreactivity is something that is often found in these highly conserved regions for polyreactivity or for universal vaccines. And so it looks like we have a lot of actually overlap with our own research interests, which is really exciting. So I'm so happy to be talking to you today. Just a little bit about Julie. Julie is a researcher at Gilead Sciences. She did her undergraduate here at UT Austin and her PhD at Stanford University. So thank you so much for talking with me today, Julie.
Yeah, Dalton, we actually kind of have connections through our academic lineage. So when I was at UT, I worked with Jennifer Maynard, who was a PhD student in Dr. Giorgio's lab. So there's some connections there. And then there's also the connection with working on a broadly protective influenza vaccine during my PhD. So very clear crossover here.
Yeah, Dr. Maynard, she's on my committee. And during my candidacy exam, I mean, she's just so nice. It was enjoyable to actually just do my candidacy exam, which is something that's supposed to be super stressful. But
I don't know if I've ever heard anyone say that they enjoyed their candidacy exams, so that might be a first.
But Jennifer Maynard is fantastic. So yeah, I guess hopping into it, I wanted to really just first get a picture of why you decided to pursue chemical engineering, and more specifically, what got you involved in therapeutics.
So you're going to find a theme when I talk about my career, both academic and now industrial, which is, I like to use this quote: I may not have gone where I intended to go, but I think I ended up where I needed to be. And as a high schooler, I was convinced I wanted to be a pediatric oncologist. I had my whole life plan. I was going to study biology or chemistry or something like that. I was going to go and get an MD. I was going to go straight into oncology. I knew what I wanted. Clearly, that's not where I ended up. And when I was looking at schools and thinking about what undergraduate degree I wanted to pursue, my dad is an engineer. And if any of you have parents that are engineers, you know that they're strong proponents of engineering as a career path. Right. So my dad was really insistent that I look into different engineering disciplines. So at the time, the thing that made sense was biomedical engineering, right? I want to go into medicine. Biomedical has the word medical in it. Right. That was the obvious choice. So I came and visited UT actually one Saturday for a Women in Engineering program event. And they required that you visit 2 departments. And I was like, well, this is super annoying because I know I want to be a biomedical engineer. So why wouldn't I just go visit biomedical engineering? But they wanted 2 departments. So I was like, this chemical engineering thing doesn't sound awful. Fine, I'll go visit their department. And when I went and I sat through the presentations, I realized that a lot of the therapeutic type research, the new drug, new vaccine type of research that was going on at UT was going on in the chemical engineering department. And this really excited my dad because my dad is a mechanical engineer and he really likes the utility of mechanical engineering. And he sees the versatility and utility in chemical engineering as well. So he was like, yeah, get excited about chemical engineering. And he introduced me to one of his previous bosses who has a PhD in chemical engineering and had her talk up how great of a field it was and how versatile it was. And you could do, 1,000 different things with it. And between that, her talking to me and talking to the Women Engineering program, listening to the presentations, I was pretty sold on chemical engineering. So it was my first choice when I applied to UT. And so that's how I ended up in chemical engineering. How did I get away from this, hardened goal of becoming a pediatric oncologist? As I got involved in research as an undergraduate, I worked with 2 different faculties. I worked with Dr. Peppis for my freshman and sophomore year, and then with Dr. Maynard for my junior and senior year. I realized that the thing that pushes medicine forward is a lot of the academic and industrial research that's going on, because that's what gives doctors the tools that they have available to them. So the doctor is only as capable as the tools that they have in front of them. And those tools were coming out of these academic and industrial research labs. And so that's really where I kind of fell in love with this idea of moving medicine forward from a research perspective, as opposed to being the doctor on the front lines.
Right.
And so there was a brief period of time where I thought, well, I'll get an MD, PhD. But the further that I went through my research experience as an undergraduate, more and more I fell in love with academic research and sort of pushing the boundaries of what's known. And I decided that a PhD was the route for me. And so that's what pushed me forward into the PhD route instead of the MD route. And I couldn't be happier. So I think it was the right decision.
Yeah, I think when people ask me about chemical engineering, I think, like you said, the biggest selling point to me is the versatility of it. I personally came in not really knowing exactly what I wanted to do. And a lot of my undergraduate research, I actually was in material science, which is totally different. But through the coursework and through my professors, I was able to really dive into the biology world and really get started with that. And then I came here to UT Austin to get it. So yeah, the versatility is tremendous in chemical engineering for sure. So so you did research here with Dr. Maynard and Dr. Pepys, and then you pursued your PhD at Stanford. Can you tell us a little bit about the projects you worked on during your PhD?
Yeah, so during my PhD, I worked for Professor Jim Swartz, and his lab is really kind of the epitome of chemi. So he has a background both in energy and in pharmaceuticals. And so his lab, when I joined, had really two main streams. So one group worked on biological production of hydrogen, and then the other group worked on vaccine development. And while those seem like completely disparate topics, they really aren't, because they're all focused on protein engineering, and for Jim's lab specifically on cell-free protein synthesis and using those techniques to drive these two different research areas forward. So I had fallen in love with immunology while I was an undergrad at UT Austin. It's sort of that really intensely unknown field. And I loved the idea that there was more that we didn't know than that we did know. So I was really drawn to the vaccine and the therapeutic development side of Jim's lab. So what I worked on was taking a virus-like particle platform. So taking a really stripped down version of the hepatitis B virus, just that core protein, this shell type structure, and using it to create a really modular vaccine platform. And what's really interesting is at the time, we talked about this hypothetical world where a pandemic disease arose and we needed to quickly generate a vaccine. And now, just a couple of years after my PhD, that is no longer a hypothetical world. That is a very real part of our day-to-day. And so the idea would be that in an instance like COVID-19 or some other pandemic disease, this platform that we had developed would be very much plug and play. So you could pull the antigen that you were interested in. So if you were talking about COVID-19, since that's something we're all really familiar with, you'd be talking about the spike protein.
Right.
You could make that spike protein really quickly using cell-free protein synthesis. And then almost like Legos, it would connect to this virus-like particle platform. Mm-hmm. And so you get this sort of plug and play type vaccine development that would hopefully be a much quicker route to a vaccine than the more traditional type of methods. And so while I was at Stanford, I worked on a Zika vaccine, broadly neutralizing influenza vaccine. So that's where we kind of tie into each other academically. And then I also did some work on some other ideas with this virus-like particle platform where maybe we could induce T-cell immunity as opposed to B-cell immunity more effectively, or potentially deliver some other types of adjuvants or immune stimulating molecules along with our vaccine platform. So I got to do a little bit of development of the platform, but also some work on just demonstrating the utility.
Yeah. So do you see, like, for the future of like a universal vaccine, is there a technology that like you think is your favorite? If you're going to guess, like, which one's going to take the lead on universal vaccines, like what type of platform do you think would that be?
I'm a big fan of the mRNA vaccines. They've really proven themselves. So I think it'll be really exciting to see, like I know Moderna was talking about potentially an influenza vaccine, also potentially talking about testing an HIV vaccine. So there's a lot of stuff in the works there. And I'm excited to see how that pans out because there's been a lot of work that's gone on into developing that platform. So seeing it have the opportunity to shine is really cool.
I think one of the coolest things about the mRNA vaccine, which we weren't really be able to do quite well with the old vaccines, is that if you were to ask someone on the street, like, how do we provide protection against as a lot of different variants in one vaccine? I think something reasonable someone might say is, why don't you just throw as many variants into a single vaccine as possible? And for traditional egg-based vaccines, there's a lot of challenges in actually doing that. But I'm starting to hear stories and trickling of people like showing 20 different hemagglutinin proteins in a single mRNA vaccine in like mice studies, and they're developing titers against all of them. So I think mRNA is sort of providing like a shotgun type approach to the universal vaccine challenge, whereas historically it's been a lot of like really smart engineering to try to target these heavily conserved regions. And I think, from my own perspective, the like targeting conserved regions can be very difficult because a lot of the antibodies that one is the immunodominance of usually the variable regions of some of these viruses, but also the ones that do bind these conserved regions often display these polyreactive and autoreactive characteristics, which it's still really unknown how beneficial or detrimental these can actually be to somebody. So it's a really exciting time to be an immunologist.
Well, and it's also just with engineering a lot of those conserved domains, they're conserved because they're well protected. A lot of times they're also conserved because they have multiple structural conformations that they can undertake, and they can be really, really tricky to engineer in a way that is easy to present to the immune system in a traditional vaccine type format. Absolutely. So they're just the level of engineering and producing a lot of those conserved domains is really complicated. Not to say that it can't be done, but some of these shotgun approaches might be a faster route to universal protection.
And so you now work at Gilead Sciences. Can you tell me a little bit about your role there and your transition from your PhD into Gilead Sciences?
Yeah, so this is another one of those left turns that I've taken in my career. So I now work in formulation and process development for the small molecule portion of Gilead Sciences. And so this is, like, in my opinion, the perfect job for a chemical engineer, because we actually get the opportunity to work with a compound early on in the research stage all the way to the commercial stage.
Right.
So we're getting to see the full spectrum of drug development while we work on our programs and our products. So what do I do in formulation and process development? This is a question that I get asked a lot. If you flip over a bottle of acetaminophen, of ibuprofen, there's an active ingredient. And that's what most people care about, is what's the active ingredient and how's it going to make me feel better? For the longest, that's all I cared about. But there's also a list of inactive ingredients, usually at the bottom of the bottle.
Right.
I used to never care about those. They had no implication on my life whatsoever. They are now my everyday. So formulation and process development is responsible for the selection of all of those inactive ingredients, developing the process to manufacture either the tablet, the injectable, the parenteral formulation, and then seeing it through all of the different stages of clinical research to a commercial product, if that is the end for that compound. And so we think a lot about scale up. We think a lot about the end goal, which is a product that you make for commercial distribution. So that means, not making 15 tablets. That means making thousands, tens of thousands, hundreds of thousands of bottles of tablets every year. And so what's cool about seeing the full spectrum from research to development is when we're working on an early research program, we can actually use that insight that we have into commercial development to help guide research in their selection of a compound. So we can identify different liabilities, like, oh, maybe this is going to be a really tricky compound because it doesn't have any solubility in any solvent that we look at. And so this is going to be tricky both from a biopharmaceutical performance standpoint, but also from a manufacturing standpoint. Or we may also be able to identify other liabilities in the compound, like the synthetic route, because we've worked with process chemists in the past on these commercial products, and we can potentially have some insight into those problems or potential liabilities early on. And so we can help identify those, and we can help guide research sometimes towards a compound that may be more developable and more likely to become a commercial drug that addresses some sort of unmet clinical need that's been identified.
Gotcha. So you really provide a really cool perspective. You get to see like the whole pipeline, as you said, for the production. Something I think that is a concern for a lot of people is just the cost of often of a lot of these drugs. And so, do chemical engineers, do we have any duty, or, what steps can we take to try to make them more accessible to people? And what are the biggest hurdles for reducing the cost for a lot of these drugs?
So one of the things that I just want to point out from the get-go is one of the things that I love about Gilead and what drew me to them is that they have a tremendous number of efforts to provide their therapeutics in an accessible manner, both in the developed and the developing world. So there are a lot of efforts to provide access to these therapeutics, both at the corporate level and otherwise. But there is a tremendous cost to making a therapeutic. And a lot of that comes from things that people don't see, which is the number of failures that happen. Drug development, you can start with tens of thousands of compounds and end up with one FDA approved commercial product.
Right.
And so a lot of what's getting captured in these costs is the cost that it takes to innovate. And so there are things that we can do as scientists, which is help identify, we call it fast to fail, right? So if a compound isn't going to work out, how can we identify that earlier on in the process so that we don't undertake a lot of the same resources to get to that same endpoint, which was, maybe this isn't the most effective compound, or maybe, this isn't the right compound, maybe it's got some toxicological side effects. How can we identify those things earlier on in the process? And you see that a lot with the research teams trying to develop the best in vitro assays early on, trying to really understand structure-activity relationship and structure-efficacy relationships. And so a lot of it is costs that are unseen to the general public because there's so much that doesn't actually come to market, so much that goes into making one great commercial product. But then also there is a lot of work that we can do as scientists on our end, both in an academic and an industrial setting, which is, like I said, to try to figure out how we can identify successful compounds earlier on, and also how we can identify compounds that may fail later.
And so is there anything currently in the pipeline? I'm not sure how much you can say for, but is there any products coming to market or currently in the development phase that you're really excited about that are really unique to Gilead Sciences?
One of the things that I really love about Gilead, and this is extremely apparent in their HIV pipeline and portfolio, is they're committed to bringing to market whatever the patient needs and evolving the compounds and the commercial products with the desires of the patient. So if you were diagnosed with HIV in the 80s and the 90s, it was really not a manageable disease. You were receiving 30 plus pills every single day. They were maybe taken with different frequencies at different times. Some need to be taken with a meal, without a meal. Some made you feel terrible, had, really difficult and hard to manage side effects.
Right.
Gilead developed the first single tablet regimen, so Atripla. And this was a game changer for HIV and HIV treatment, because these patients who are used to taking a handful of pills across a full day are now taking a single tablet once a day. Yeah. And this is a huge deal. And you could see a company saying, okay, we fixed it, great, we're done. But Gilead has continued to innovate on that single tablet regimen to make a more effective single tablet regimen, a single tablet regimen that works for a different population of HIV patients, a single tablet regimen that's smaller, so it's easier to take, because with HIV, compliance is a huge deal, and compliance is really important in managing the disease successfully. And so if you make a smaller tablet that somebody's more likely to want to take every day, it's easier to take, then that's going to be they continue to patients about, what do you want in your HIV treatment? What's holding you back from being compliant, or what would make it easier for you to be compliant with your medication? Right. And so some of the things that are, on the horizon is, could we reduce this from once a day to once a week? Yeah. Could we develop an injectable formulation that is once every month, once every 3 months, once every 6 months? And for a lot of HIV patients, this is extremely attractive as an option. And so just constantly thinking about, yes, we have a drug that's good. Can we make a drug that's better or that matches the patient's lifestyle better? Is something that I think is really cool. And I think there were a lot of points along the way where Gilead could have stopped and said, we've got a great HIV medication, a therapeutic, we've got a PrEP medication, so we're good. But instead they've said, how can we continue to push this forward and make it easier for HIV patients to manage their disease? And I think we see this in a lot of different areas that Gilead is interested in developing medicines.
Absolutely. Yeah. And my own personal experience, when I first joined the lab, I had a couple months of normalcy, and then the pandemic started. And then the McClellan lab here at UT was one of them developed one of the most stable structures of the spike protein. And so one of the things I really started doing, at the start of the pandemic, was just producing the spike protein for everyone all over campus to use for their research, try to understand, understand the coronavirus as it was developing. But I think something that was really cool about that is I would call my parents and I said, hey, I'm producing the spike protein, expecting them not to know at all what I was talking about. But during the pandemic, everyone sort of has some understanding now about the therapeutics world and immunology. And it makes it really exciting that, we can talk very easily with a lot of people about the benefits of therapeutics and how these technologies are developed. So it made it a lot easier to communicate with people for sure about what we do. I feel like, did you feel something similar as well?
Yeah, and I think one of the things that was interesting about the pandemic is science communication came to the forefront. And so even when you think about things like mask wearing or, do you need to disinfect surfaces, all of those sorts of things, right, became really hot topics, and people were really interested in the science behind, do we need to disinfect? Do we need to wear a mask? Do we need to wear masks inside or outside? And so I feel like science got this, spotlight on it, which was really exciting because, people were asking me for my thoughts on all sorts of different things. And I was like, well, this is not really my area, but I'd love to learn with you, right? Let's read up on masking and why it may or may not be effective. And that was really kind of fulfilling, was to see a lot of my friends and family have this interest in science and understanding, the things that impacted their day-to-day lives. Right. Yeah. Part of it, I feel like it's often a trope of at least doing a PhD is that you working on something and you explain it to someone, you're like, I have no idea what you're talking about. But this is definitely one of those things where everyone's has something to say about it, which just makes it really exciting to be part of the science itself. So something else I wanted to ask you about working at Gilead is I imagine that you work heavily with a lot of biologists, pharmacists, a lot of different people with different perspectives. What do you think the chemical engineering perspective brings to the table that's unique compared to some of these other disciplines? Yeah, so even within my department, so while we touch a lot of the different and work with a lot of the different disciplines across campus, even within formulation and process development, we have a lot of different disciplines. So we have people with pharmaceutical sciences backgrounds, people with chemistry backgrounds, people with chemical engineering backgrounds. And we find that we all have niches pretty quickly. So early research, there's a lot of thinking about the innate chemical properties of a molecule and understanding those better. And so early on in the research phase, the chemists have a lot of contributions that they can make because they're used to thinking really in depth on the molecular level about a compound. When you start to transition to some of the process development, you start thinking about scale up, you start thinking about the different critical process parameters, you think about design of experiments. That's where the engineers really shine, because that's something that even if we haven't specifically thought about a tablet process or an injectable process, we're thinking about scale up throughout our entire education. And so I think you start to see people develop in their expertise, but then kind of like I mentioned before, everyone's always willing to help each other out. So early on in a research program, if I'm running into something tricky about a compound, I'll reach out to one of my friends who I know is really strong in chemistry. If somebody who is working on a development program is starting to run into some tricky things with scale up, they might reach out to me or one of the other engineers in the department and say, hey, I can't really figure out what's going on here. Can you help me think about this problem? So even though we all have our little niches, we can all still work on all of the different areas, from research to commercialization, and just lean on each other for help when things come up.
Awesome. So something else I wanted to ask is, having been part of Gilead for a few years now, talking to someone who will likely be going into industry in a few years, what is something that you wish you knew when you were sort of, in my perspective, or even an undergraduate, that you know now that you wish you knew then?
I think I touched on this a little bit, but just to be open to all sorts of opportunities. So if you had asked me, if you'd asked me when I was an undergraduate, if I would work in small molecule formulation and process development, I would have said, probably not. If you'd asked me halfway through my PhD, I didn't really know what it was, but I didn't think it was, whatever I thought it wasn't what I thought I was going to do. Yeah. But being open to the opportunity when someone from my lab reached out and said, hey, my department's hiring, I think you should interview, I think it'd be a good fit for you, and saying, okay, sure, why not? Let's see what this is about. And I've really gotten a tremendous amount of experience. I've learned a tremendous amount. This department puts you in a place where you learn every single day, and every single day is something a little. So I think being open to new experiences, new opportunities, things that might feel a little bit outside of your comfort zone or the area that you've really focused in on in your PhD is a really important thing, because you never know what opportunity is going to take you to that next level, and be something that's really exciting and something that you enjoy doing every single day.
Right. Yeah, absolutely. And so I guess spinning off of that, what do you think is the biggest challenge that the therapeutics industry currently has that we need to address? Or I guess if you had a control over, what the therapeutics industry was doing, what type of areas would you focus on yourself?
That's a tricky question. I'm going to spin it a little bit.
Okay.
One of the things that I think is really interesting that I think would be really valuable to general therapeutic development is a lot of the things like organ on a chip, where you're thinking about taking some of these experiments that we currently do in vivo and making them an in vitro experiment that may even be potentially more representative of, a human metabolism of a drug or metabolism of a compound.
Right.
And so that's something that I think is really exciting, and it's something that a lot of academic labs are working on. And I think it'll be really cool to see how they progress forward, and if they are able to develop organ on a chip that are truly representative of the human body and that could be used in place of a lot of these in vivo experiments that we currently run. That could be something that would be game changing, both from just a animal use perspective, but also from a cost perspective, right? An in vitro experiment is always going to be cheaper than an in vivo experiment.
Right.
So that's something that I definitely have my eye, the corner of my eye on, looking at academic going on. I think that's something that's really cool and has a lot of potential.
Yeah, we just have a new student in our lab who actually does a very similar thing, monitoring the intestines, basically, that are absorption of nutrients and stuff, using organ on a chip like device and stuff. And we previously used the Mimics system, which is similar to a chip on device. models like a lymphocyte, so you can like do affinity maturation on some antibodies in an artificial setting. So yeah, a lot of really, really cool technologies for in vitro applications for sure. And so I guess we're approaching the end here. I just want to leave off on more some fun questions. What's your favorite part about, being in therapeutics?
I think I love that every day is different. And part of that is specific to being in formulation and process development. So I love that every day I get to tackle some new challenge. And even within a day, you can spend a morning working on, early research. And then in the afternoon, you can be working on, a late stage clinical compound, which is really cool. So it's constantly keeping, those brain cells moving and thinking about all of the different attributes of a compound that make it a commercializable drug.
Right.
The other thing that I really like about being in therapeutics is just that we are on the cutting edge of medicine. Gilead's tagline is that we're creating possible. And so there may be things that we all think are impossible today, that, a disease that we think may not have a cure, or some other therapeutic innovation that we, can't even imagine right now. But one day in the future, somebody is going to create and bring that therapeutic innovation to realization. And so it's just really cool to kind of feel like you're sitting right on the edge of something that people never even imagined.
Absolutely. And I think people tend to forget just how young immunology actually is, and just, how recently, some of the very basic concepts of immunology was just discovered like a couple decades ago. This isn't a very old field. This is all very new. So it's exciting to be at this almost like this exponential growth of the field right now. But yeah, with that though, thank you so much for chatting with me today, Julie. This was really great, and I loved hearing your perspective on things, and really excited to hear about what Gilead Sciences is doing next.
Of course, thanks for having me, Dalton. It was great to meet you.
Thanks. Thanks.